Welcome, folks, to part 3 of chapter 11. We're going to continue our discussion of prokaryotes. Specifically, we're going to be talking about gram-negative bacteria. So we had already talked about archaea, we have talked about our gram-positive bacteria, and now we're going to talk more about our gram-negative bacteria. Just a reminder that gram-negative bacteria, they don't have a thick peptidoglycan cell wall like gram-positive bacteria do, but they do have a secondary cell membrane on top of, so they have their cell membrane, then they have their thin peptidoglycan cell wall, then they have this secondary cell membrane. And this cell membrane has some unique characteristics and specifically has some components in it that our immune system reacts very specifically to. And so some of the pathogens that we talk about in our lecture today, just because they're gram and they have these components, these LPS membranes and a lipid A component, they are actually pathogenic. Those components are pathogenic and are recognized by our immune system and are responded to by our immune system. And so as we kind of go through these gram-negatives, you will see some of that. So the gram-negative proteobacteria are categorized into 6 similar or related groups. And so you have them just, organized as alpha, beta, gamma, delta, epsilon, and zeta proteobacteria. And really the delineation of GC content is not relevant in this category in gram-negatives, so we just kind of talk about them in these related categories here. So we'll go through these 6 related categories and talk about some of the representative organisms within. So alpha-proteobacteria typically are aerobes that are capable of growing at low nutrient levels, and so they have in them some unique metabolisms. So nitrogen fixers, for example, or hydrogen gas production, or nitrifying organisms, or phototrophs. And so they kind of tend to have organisms within them that have unique metabolism, although there are some pathogens within them as well. And one thing that's characteristic about alpha-proteobacteria is that many of the species within produce what's called prosthecae, which allow them to adhere to surfaces. Since they have these unique nutrient requirements, oftentimes they need to adhere to surfaces where those nutrients are present. And that's also important in the production of various biofilms. So you can see the prosthecae here. Instead of it just being an external kind of attachment like a filament, it's actually almost an extension of the cell that actually has some cytoplasm within it, and it actually allows them to attach and adhere to various surfaces. So within the alpha-proteobacteria, you have nitrogen fixers like Azospirillum and Rhizobium. Azospirillum associates with the roots of different plants, and it actually fixes nitrogen within the soil, and it stimulates root hair growth as well. As it's fixing that nitrogen, then the plant actually will grow more root hairs. Rhizobium we've interacted with a little bit in this class because we're culturing it in the garden, and it actually has a symbiotic relationship with pea plants and legumes and lives within these nodules that they produce. You can see some of those here with our nodules formed here, and the bacteria are living within those. And in this case, you're looking at the results of Azospirillum being in the soil around the sugarcane plants, where the root growth was actually stimulated by the presence of those bacteria. Here you can see in a little bit more 3D what some of those nodules look like. Okay. Here's a pea plant, and then you can see these components or compartments are the nodules on the root, see all of these. Each of these is actually hosting root nodule bacteria that are doing nitrogen fixation, and then that plant has access to them. So also in the class Alpha-proteobacteria are purple non-sulfur bacteria, which we have talked a little bit about already. And these are phototrophic, and they live kind of like at the top of mudflats in shallow aquatic systems. Okay. There are some pathogens in alpha-proteobacteria as well, specifically Rickettsia and Brucella. Rickettsia is an obligate parasite. So it's a gram-negative, it's aerobic, and it's rod-shaped, and it obligatorily has to live within the cells of or within another organism. And so generally it's living and reproducing within mammalian cells, but it actually can be transferred in a vector, specifically arthropods, vectors that are arthropods like fleas, ticks, mites. And it causes rickettsia, rickettsial species cause typhus and Rocky Mountain fever. And so that can produce a rash, muscle pain, and other symptoms as well. Brucella is a common pathogen of mammals, and it causes chills, sweating, fatigue, and fever in humans due to its pathogenic abilities and to be able to evade the human immune system. Other interesting alpha-proteobacteria include Acetobacter and Gluconobacter, both of which produce vinegar naturally. And then also Caulobacter, which produces the strongest adhesive known to mankind. Its primary function for the bacteria itself is to adhere itself to surfaces to make biofilms, or to adhere itself to clusters of Caulobacter cells so that they can exist together in community. But the adhesive is so strong that it could actually hang an elephant, an adult elephant, from the surface of a quarter, the coin, like a money coin. So the surface of a quarter would be enough to hang an adult elephant from. So very strong, or something the weight of an adult elephant. So a very strong adhesive, and you can see here that the purpose of that is to produce biofilms or to exist in colony. There's another alpha-proteobacteria called Agrobacterium that's really interesting from a lab perspective, because what it does, and actually you're probably anecdotally familiar with this bacterium, because you might see trees in the forest or in the city that have these tumors. And what's happening is the bacteria actually infects the tree, and then through a pilus, it actually inserts genetic information into the plant cells that trick it into making these tumors. And so the tumors provide nutrients for that bacteria and additional space and growth for that bacteria. But it's very interesting because this is an example of natural transgenic DNA transfer, where you have one domain separate bacteria then transforming the DNA of a plant, which is in eukarya, a completely separate domain. And so from a genetic modification perspective, being able to take genes and insert them in different organisms or even different species or domains is a useful technique or procedure. And we'll actually do that in the lab, where we'll transform an E. coli with a gene from a jellyfish, a fluorescence gene from a jellyfish. And so looking at some of these natural systems like Agrobacterium, where this is happening in the environment, and transfer of plasmids between different bacteria and so on and so forth, where we have these examples in nature, allows us to monopolize on these processes in the lab as well. Then we'll move to class Beta-proteobacteria. Beta-proteobacteria is a diverse group of gram-negative bacteria, and they thrive in different habitats. Some of them do thrive in habitats with low nutrients, and even though they're genetically different, they have some overlap with alpha-proteobacteria as well. So you'll see that we have soil bacteria that are nitrifying. We also have Neisseria. Neisseria is gram-negative Diplococcus, and that is a natural inhabitant of the nasal cavity and urogenital regions of mammals, and a natural part of the microbiome, but it can actually release toxins, and it can be, some species can be pathogens. And so specifically Neisseria gonorrhea, which is a sexually transmitted disease, and Neisseria meningitis, which is actually the most common cause of meningitis in younger adults. So if you're in a clinical setting and you have a younger person that's coming in with a meningitis infection, it's likely Neisseria meningitis. Then we have Bordetella. Bordetella is the cause of whooping cough, or the causal agent of whooping cough. Whooping cough has decreased in its incidences in the US because of the vaccine DTaP, which we had mentioned already. So DTaP is diphtheria, tetanus, pertussis. Pertussis is the name for whooping cough and polio. And so it protects against these different diseases and has decreases the likelihood or the cases of whooping cough in the US. Then we have Burkholderia. Burkholderia is interesting because it actually is able to recycle a number of unique chemicals in the environment, specifically soil contaminants like PCBs and other chemicals, but it also can colonize the respiratory tracts of patients with cystic fibrosis, and it can be pathogenic in some cases as well. And it can develop, or there are strains that are highly antibiotic resistant. So depending on the strain, depending on the environment, kind of have this various characteristics of Burkholderia. Other beta-proteobacteria include thiobacillus, which is able to recycle sulfur in the environment, and then a number of different genuses that are able to, that are relevant to sewage treatment. Either they're helpful or harmful. And so essentially these two genera here, Zooglia and Sphaerotilus, both make what are called flocks. And flocks are essentially these slimy masses similar to biofilms that are aggregates of both bacteria and, and organic matter. And so those flocks, if they allow for you to separate out organic matter from water that's being, purified in water purification systems, then they're helpful. But if they don't separate out, they can actually block pipes and filters in water purification systems. And so you can see kind of two different distinct types of flocks. So here we have Zooglia, which has flocks that actually, they actually kind of sink to the bottom and allow for water for purification, whereas Sphaerotilus, actually the flocks mix within the water. They don't sink down to the bottom, and they can actually block pipes, filters, so on and so forth. Then we have our Gamma-proteobacteria. Gamma-proteobacteria are the largest and most diverse of the 6 groups of proteobacteria, and we can divide them into these 5 categories. We have sulfur bacteria, which we have talked about before, and so you can see some sulfur bacteria here, and actually sulfur bacteria will sometimes make symbiotic relationships with sulfur worms. And I heard, a really interesting piece on sulfur worms and sulfur bacteria in an NPR shortwave recently. So I'm including that link, the first link in this part below. If you're interested in listening more about that, I think it's about 14 or 15 minutes. Then the second category within Gamma-proteobacteria is intercellular pathogens. So examples include Legionella and Coxiella, and both of them are actually able to live within the cells of the immune system. They're intercellular pathogens within the cells of the immune system, and they're actually able to evade the immune system that way by living within them. Legionella is also an intracellular pathogen of amoebas, just a fun fact there. The third category of Gamma-proteobacteria are methane oxidizers. And so these are organisms that, instead of producing methane, are actually metabolizing methane. And it might not surprise you that they tend to live right above methanogens. So if you remember back to our discussion of archaea, they tend to live right above methanogens in mudflats and aquatic systems because they will actually take the methane that methanogens are producing and then metabolize it. Then we have glycolytic facultative anaerobes, and these are essential within the microbiome for a number of reasons. Essentially, the group as a whole tends to be able to break down various types of carbohydrates through glycolysis. And since our digestive tract makes so many of these various carbohydrates as it's breaking down the food that we eat, there are a number of organisms within this category that are part of our microbiome. But there are also a number of organisms within this category that can infect our digestive tract because of all of those carbohydrates that are produced and actually produce pathology and infection. And then you have some organisms that are actually natural parts of the microbiome that might be opportunistically pathogenic. Okay? And so you have a spectrum. So in terms of organisms that are naturally part of our microbiome, like Escherichia coli and Klebsiella, and these are also opportunistic pathogens. And then you have true pathogens, or organisms that shouldn't be in the microbiome. And if they are there, they're usually inducing pathology. And so Salmonella, Shigella, Yersinia pestis, so one of the species within this genus is actually the cause of the plague. And so a number of relevant organisms within this category here, specifically to the digestive tract. Okay. We also have pseudomonads. Pseudomonads are also able to break down a number of organic compounds within the environment, and some of them are nitrogen fixers as well. These tend to be aerobic, flagellated, either rod-shaped or vibrio, so curved rods, shaped organisms. And the ones that are able to recycle various environmental components are able to recycle some really strange and odd components, like rubber, oil, even plastic and TNT and other hazardous byproducts. The problem is sometimes when they recycle those hazardous byproducts, they themselves generate hazardous byproducts. So they can be useful in some regards, but then also we have to be thoughtful in how we use them. They can also be pathogens of humans and animals and are specifically involved sometimes in the spoilage of refrigerated items and can live at slightly lower temperatures, like refrigerated milk, eggs, and meats. Okay. But there are pseudomonads that are also part of the natural microbiome. Then there are also nitrogen fixers that are soil dwelling in this category, like Azobacter and Azomonas, both of which are within the soil, but they don't within the soil as nitrogen fixers. But the distinction between these guys and previous nitrogen fixers that we've talked about is that they don't specifically adhere to the roots of plants. Okay. The predominant genus within Pseudomonads is actually Pseudomonas, the genus Pseudomonas, which has these very characteristic polar flagella as well. Then we have a couple of smaller classes within proteobacteria, so delta proteobacteria, but they do include some interesting genera. So disulfovibrio is able to actually reduce sulfate to hydrogen sulfide, and therefore it can recycle sulfur in the environment. It can live in various types of polluted streams, sediments, sewage lagoons, so on and so forth. And it can be the reason why some of those bodies of water have such a bad smell. It can also corrode. It does produce byproducts that corrode piping, sewage piping, and things like that. So important within those systems. Then we have this Delivibrio, which is an interesting because it is actually a gram-negative pathogen of gram-negative bacteria. And so it's a very small organism that's able to actually infect the bacteria and then live within the intramembrane space, live there, metabolize, reproduce until it essentially takes over the organism from within that intramembrane space. And then it has an interesting way of reproducing in that it elongates really long, kind of like with binary fission, but even longer than usual, and then divides into multiple daughter cells. Okay. Then we also have Myxobacteria. Myxobacteria had previously been thought to be a slime mold, which is a fungus. But upon more interrogation in terms of their genetic sequence and also their cellular structure, they're definitely prokaryotes, but they're very unique in that they're some of the only prokaryotes that have cell differentiation, cell-to-cell hormone communication, and group movement. So they actually kind of act like a small multicellular unit, even though they are not. It's just a colony, but they're kind of able to interact in that way. They also have a very large genome, which might be expected from an organism that has more characteristics or more abilities to interact with adjacent microorganisms. So their genome is about 10 million base pairs long in comparison with something like E. coli, which is just under 5 million base pairs long. And so they're also so cute. I mean, just look at that. Structures that they make. And as they're kind of coming into those aggregates and then they have this cells differentiation, they can also move or slide as an aggregate. So they're very unique in many ways. These are often coprophilous. Coprophilous. So as an optional addition to your discussion post for this chapter, what does coprophilous mean? Maybe if some of y'all are into anthropology or fossils, you know what coprolite is? Coprolite. So what is coprophilous? Okay. So I'll let you add that there. Okay. Another class of proteobacteria is Epsilon proteobacteria, and some specific genera within are Campylobacter and Helicobacter. Campylobacter can cause poisoning of the blood and inflammation of the intestines. And Helicobacter is one of the causes of ulcers within the stomach lining as well. And then the last but not least, well, last and smallest, so I guess in a way least, but very interesting, is a class called Zeta-proteobacteria. And these are very recent in our kind of understanding of and kind of adding to them. So the first discovered sequence within this class was discovered in the mid-2000s. So very, very recently. It's common for these to be marine organisms, and there's only about 20 identified species, and only a few of them have actually been named so far. They tend to be autotrophic. They tend to use carbon dioxide as their carbon source. And so one of the organisms that you see here is Mary profundus ferrooxidans. And so actually the name is very telling, Mary meaning sea, profundus meaning deep, ferrooxidans oxidizing iron. Ferro is in reference to iron. And so these are organisms that live in deep sea vents and oxidize iron. And they are just beautiful. They actually have different phases. They have a free living motile phase and then a phase where they're actually attached to various surfaces with these stalks here. And so these stalks are twisted filaments of iron oxides and organic material. And then you can see the cell here. So here's a cell, and here's these beautiful stalks that they use to adhere to surfaces. So really, really beautiful organism, but still we're learning a lot about this category of organisms here. Some other gram negatives. Essentially, there are a couple of phyla of gram negatives that just don't seem to fit within those other groups that we have mentioned so far. They're, from a genetic perspective, they just don't align very well with any of the other groups. And so they're kind of included separately together, not because they're relatives, but just because it's basically convenient to separate them from the other organisms that they're obviously not related to. And so these include chlamydia, spirochetes, and bacteroids. Okay. So starting with chlamydia, they tend to be very small, gram-negative cocci. Some are actually smaller than the largest viruses that we know of. But they are more complex than viruses, and they do have their own metabolism. So they are bacteria, but there's some that are smaller than the largest viruses that they know. And chlamydia is actually the causal agent of sexually transmitted disease, the most common sexually transmitted disease in the US. Okay. Then we have the spirochetes, a number of which have interesting capacity, and these are the spirochetes it refers actually to that shape where they have like this long helical structure that's very flexible, and that actually allows for them to bore into their host. And so there are a number that are relevant to pathology. So you have treponema, which is the causative agent of syphilis, and Borrelia, which causes Lyme disease. And so I've actually provided a video that allows you to see Borrelia in action. It shows you how it's able to penetrate into its host, and also some of the ways that it evades the immune system. And then I've also included 2 optional videos. One looks at how Borrelia, the spread of Borrelia, is connected to climate change. And then the second one is an acknowledgement that a lot of what we know about treponema, which is the causative agent of syphilis, has roots in racial and social injustice. And so that's about a 15 to 17-minute watch. So it's an optional watch, but I do recommend kind of considering that history. Then we have Bacteroides, which are common inhabitants of human digestive tracts, human and animal digestive tracts. They tend to be obligate anaerobes, and they can metabolize a diverse number of carbohydrates, which is why they're so important to the digestive tract. And in fact, depending on the person and the ecosystem that they live in and so on and so forth, between 5 to 50% of the species that are isolated from feces are actually Bacteroides. Okay. Although most of them are beneficial, there are a couple species that are pathogenic as well. And so they are actually, those species are some of the most common anaerobic human pathogens that we know of. Then you have Cytophaga, which is which is an aquatic species that is able to break down various carbohydrates and is relevant because it will actually break down man-made structures within aquatic settings, like the hulls of boats and piers and things like that. Yeah, those artificial structures, those man-made structures, it'll break those down. And so what we've done here in the 3 parts, that's the end of part 3, essentially. What we've done here in these 3 parts is we've talked about the different categories of archaea and bacteria. We've talked about deeply branching bacteria. We've talked about gram-positives and gram-negatives, and we've talked about some representative organisms within them. And we've made some connections between organisms and disease, causation of disease, environmental impacts, so on and so forth. And so that basically concludes our discussion of chapter 11. I would recommend that you take a moment to pause, review your notes, look through your different, diagrams or concept maps that you've made. And in terms of what you need to know for this chapter, look at the learning objectives, because what we've done is a survey in lecture, but there are specific organisms that I want you to focus on for the assessments for this class, and you'll find those in the learning objectives for this chapter. Thank you for listening, and I will see you in the next series of videos, which will be on chapter 13, where we'll look at the microbial agents, viruses, and prions.